know that in 1953, Watson and Crick, they determined the structure of the DNA, double helical structure. And the first sequencing, the modern sequencing, actually, it started in the 1977 with the discovery of the chain termination method and the DNA degradation method. That is called Sanger sequencing and Maxam-Gilbert sequencing. But over the period of time, of course, because of the chemical composition and the hazardous chemicals used in the Maxam-Gilbert sequencing and the tedious nature of that sequencing, that couldn't stand over the time. But the Sanger sequencing actually ruled the science field over next 20-25 years, and most of the sequences were done with the Sanger sequencing only. And you know that in 1990, actually, Human Genome Project started. So it was the Sanger sequencing only which was used for doing this. And it took around 11 years to first draft completion. And finally, in the 2003, the first completion of the Human Genome Project was published. And it means 13 years and $2.7 billion were expended on this particular project. So because the first generation, oh, I forgot to, we have to record also, right? So just a moment, I'll put the recording on. Anyway, I don't know where it is. Anyway, so yeah, so this Sanger sequencing method actually is called as the first generation sequencing. And after that, whatever the sequencers they were discovered, they are called actually as the next generation sequencing. So in the Sanger sequencing, initially when it started, there was no PCR as such, because the 1977 there was no PCR. So for amplifying the DNA were cloned in vivo in the bacterial DNA, in the plasmid and all. So there was for the amplification, in vivo amplification, plasmids were used. So that was quite tedious. And of course, the first generation sequencer or the Sanger sequencing, actually, it was, you cannot generate a lot of data. And it took, of course, you know that 13 years it took for the human genome sequencing, and now we can do in a day. So scientists started thinking of a new technologies, and finally they discovered in 2005 a new approach for sequencing the DNA, and they all are called as the next generation sequencers. So just a minute, some people are waiting in the room, so I'll just admit them. So next generation sequencing, actually, they are faster than Sanger sequencing, of course. They can massively parallel sequencing can be done, and millions of bases at one. I mean, the parallel sequencing means millions of bases are sequenced at a time, and they produce enormous amount of the data which can be used for a lot of things. So as I mentioned, there are first generation sequencers. So first generation sequencing is, of course, the Sanger sequencing, and after that, whatever the sequences they were discovered or invented, they have been categorized in the second generation, third generation, and the fourth generation sequencing. So as I mentioned, sorry for, I have to admit few people. So second generation sequences are actually the first one which was commercialized was the Roche sequencers, and which is based on the Pyro sequencing. And it started actually in 2005. So second generation sequencer, actually what they did was they, because in the first generation there was a cloning part, But in the second generation, they remove that cloning part, and now there is a PCR-based enrichment of the DNA. So either emulsion PCR or the bridge PCR. So in the first PCR, in the first generation, there was the main difference in the first generation and the second generation is actually the PCR-based. Second generation, they are PCR-based, and first generation, they were cloning-based. So I have just listed few. I'm sorry, in between, I have to admit few people. So, and then comes the third generation sequencer. So third generation sequencer, actually, they also remove the PCR-based based amplification of the DNA. Now in the third generation sequencer, only a single DNA molecule is sequenced. So there are Pacific Biosciences and the Nanopore sequencers. And the fourth generation sequencers are actually, they don't even single nucleotide, but even the single cell or the tissue. They sequence the DNA from the single cell and the tissue. So these are the main differences of the first generation, second generation, third generation, and fourth generation. I hope I'm clear to everyone. So basically, for the library preparation in the next generation sequencing, there are three different steps. First is, of course, the fragmentation. Whatever the DNA you isolate from whatever the organism, you have to do the fragmentation. That fragmentation can be done either by the enzymatic method or through the mechanical method. So you fragment the DNA in the small pieces, and then you ligate the adapters. So these adapters are different for the different sequencers. This is just for the Illumina sequencer I have presented. So you have to ligate the adapters. These adapters are actually complementary to the flow cell oligos. And then finally, we do the amplification to enrich the DNA to make more DNA. So these are the basic steps in the library preparation. For the second generation sequencers, you have to fragment the DNA, you have to ligate the adapter, and you have to amplify the DNA. This amplification can be either through the emulsion PCR or through the bridge PCR. So the first sequencer which was launched, actually the next generation, is the Pyro Sequencing. The Pyro Sequencing, the name comes from the pyrophosphate. So in this case, of course, we do the fragmentation. As I mentioned, that the first step is you have to do the fragmentation. So we do the fragmentation of 400 to 600 bases, and then we do the emulsion PCR. So emulsion PCR is actually, there is a Cephalous beads, and these Cephalous beads actually they have the oligos, which is complementary to your DNA adapters. These adapters, they are complementary to these beads oligos. And what we do in this case is we dilute our DNA samples and the beads in such a way that one bead can have only one DNA fragment. So you can see over here, one bead is there and one DNA fragment. That dilution we have to make. And then in a tube, of course, we put these things with the emulsion oil. So what oil does is, and we do the centrifugation and all these things. So what happens is there is a droplet, one droplet forms, and with that one droplet, there is one Cephalous bead and one DNA, and all the materials required for the amplification. So see here, microtiter plates are there, and then amplification starts. So it means that on one bead, the amplification happens, and in that bead, all the amplified product are from the same DNA fragment. And then, of course, finally, we break that emulsion, and these emulsions, they are loaded into the microtiter plates. So you can see that these beads, amplified beads, where a lot of DNA fragments are there of a single DNA fragment, and then they are loaded into the microtiter plate, and then the sequencing starts. So in this sequencing, actually in the pyro sequencing, what happens is the annealing happens with the primer, and then one by one, the nucleotide gets added. Let's say suppose first time A comes in. If there is a complementary sequence of A is there, let's say suppose here A is there, so the complementary of A is T. So if A is coming, it won't be added. When T is coming, it will be added. So when it's added, you know that during the reaction, polymerase reaction, the pyrophosphate part gets released. So when the pyrophosphate parts gets released, there is one enzyme called APS, which converts the pyrophosphate into the ATP. Just a moment. So ATP, and then this luciferase enzyme, it converts the luciferin into the oxyluciferin, which emits the light. So it means that if there is addition of a nucleotide, pyrophosphate will be released, and then this pyrophosphate will finally gets converted into the light. So if the light is emitting, it means that the DNA, the one nucleotide has been added, and that's how the sequencing proceeds actually. So this is the basic concept of the pyro sequencing. But the problem with the pyro sequencing is it is quite expensive, like $10,000 it takes for the billion of bases. Lower read accuracy. So read accuracy, so read over here, just I wanted to mention one thing, that the read means one DNA fragment which gets sequenced. That is called read in the sequencing term. So the lower reading accuracy, it means the sequence, we are not confident enough to tell that, okay, this particular nucleotide is accurate. So there is a lower accuracy of the reading. And the signal strength for the homopolymers after 8 nucleotides, they are not that accurate. The meaning that, you know, that there are poly-A tail, or there are sometimes stretch of the nucleotides which are repetitive. So when it comes to the repetitive DNA fragments, these pyro sequencing actually, they do not give the enough signals to be accurately those reads can be sequenced. So that's the big problem with the pyro sequencing. And so the next one is ion torrent. So ion torrent, actually the same person, the Rothberg, actually he was the main person who actually started the pyro sequencing, and then he left that company, Roche company, and started his own company and started a new sequencer that is called ion torrent. So that's why I put it pyro sequencing in the ion torrent together. But this ion torrent was first started actually in 2010. So in this case, the basic principle of the library preparation is exactly the same. You have to fragment the DNA, you have to align you have to add the adapters, and then you have to do the amplification through the emulsion PCR. So, but the biggest advantage of this particular sequencer is ion torrent is it is semiconductor based. So there is no fluorescence signal in this. So what happens in this case, as you all of you know, that when the polymerization of the DNA fragment happens, one nucleotide gets added, and when the one nucleotide gets added, there is a release of the hydrogen ion happens. This release of the hydrogen ion actually causes the change in the pH of the solution where the reaction is happening. And this change in the pH causes the change in the voltage. This voltage, change in the voltage is recorded, and which is now finally converted into digital form that is for your sequencing. So let's say suppose your DNA is there, one by one, the DNA fragment nucleotides are getting added. Let's say for the G, C gets added, one hydrogen ion gets released, and then this release in the hydrogen ion causes the change in the pH of the temperature, and that change in the pH of the temperature pH is 0.02. So when one hydrogen molecule or hydrogen ion gets released, there is a change in the 0.02 pH. This change in the pH, actually, there are sensors, so there is change in the pH, and then there is a change in the voltage. When the change in the voltage happens, then it say that, okay, now C has been added, so it will record that one nucleotide C has been added. Then similarly for the, once that part is done, then all the 4 nucleotides, they will come and they will match. Whether if it is G is coming, of course it won't match with the A. Then C is coming, it won't match with the A. And if T is coming, of course it is complementary to the A, it will attach over here. Again, hydrogen molecule will be released. There will be change in the pH, change in the voltage, and the next nucleotide will be recorded. That's how the ion torrent actually works. So this is what I was talking about, like different molecules, DNA nucleotide molecule, A, T, G, C. For each cycle, all the 4 nucleotides, they comes in, and whatever the complementary nucleotide is there, that will be added. All other will be washed away, and the hydrogen molecule will release, and that hydrogen molecule actually causes the change in the voltage, and that is finally recorded in your sequencing. But again, the problem with this ion torrent is also the homopolymer sequences. If there is a stretch of repetitive sequences A, T, or G, or C, then again it fails to sequence those homopolymers. So I'm not talking right now. At the end, I'll talk about the Illumina sequencing, because more detail I'll talk about that. So the next sequence, so whether it is ion torrent and the Roche, or even the Illumina sequencing, they are called second generation. So in this case, there is a PCR amplification, either through the emulsion PCR or through the bridge PCR. But the third generation sequencer, that is called either Pacific Bio sequencers or the Nanopore sequencer. So there is no need of doing the PCR. So in Pacific Bio Sciences, how the library is prepared, of course, you have the DNA samples, you have to shear them, or you have to break them into the small fragments. And then again, you have to add those to the end of the DNA. There is a circular hairpin loop type structure. These circular adapters, they are get added, and then they are get sequenced. So in this case, again, you have to take the DNA samples, you have to fragment those DNA samples, and there is no PCR step in this. And then you have to add the adapters. How this Pacific Bio Sciences sequencing happens actually, so it is called smart sequencing. Smart means single molecule real-time sequencing. It means in this case, single molecule, it gets sequenced. So in this case, what happens is there are zero mode waveguides, means ZMW. These ZMWs are actually, they are one chamber which is in 100 nanometer of the aperture, and in these sequencing, the DNA there is a DNA polymerase enzyme. So this DNA polymerase enzyme is attached to the walls of this nanometer chamber. When the DNA molecule comes in, the polymerization happens. So one by one, G, T, A, C, they will come. And if the complementary sequences are nucleotides are there, they will join. And once it is joined, because each of these nucleotides, they are fluorescently labeled with the different colors. So if the G is coming, and another thing about another speciality of this smart sequencing is, instead of the fluorescence label, which is in general attached to the bases, nucleotide bases, this fluorescence in the smart sequencing is attached to the phosphate group. So you know that when the sequencing happens, when the polymerization happens, phosphate group gets released. So one nucleotide gets added, then this phosphate group will be released. Once this phosphate group gets released, illumination happens, and this illumination happens within a small chamber of jeptoliter. So that volume of that chamber is in jeptoliter, means 10 raised to power minus 21 nanometer. It means it's a very small chamber where only one nucleotide can be detected, even if there are many nucleotides outside which are coming and going, but they cannot be detected because the chamber is very, very small, which is just for the one nucleotide, and that's how the sequencing in this case happens. So in this case, smart sequencing is a single molecule of the DNA you can sequence. There is no need of doing the PCR, because what happens in the PCR is there sometimes, because the PCR biasness, wrong nucleotide gets added. When we do the sequencing, of course, the enzyme also has some fidelity issue. It cannot add all the time all the correct bases. Sometimes it's at the wrong basis also. And when we do the PCR over the 30 cycles, then there are more chances of error. So that part has been removed in this case. That is one of the advantage of the specific biosciences. Another thing, another advantage is actually in this case, you can sequence a very long DNA fragments, 10,000 to 15,000 bases. This is even, we can sequence like even bigger sequences depending on how big your fragment is. So very long fragments you can sequence with the specific biosciences. And these long fragments, advantage of these long fragments are when you do the assembly of the genome. So longer fragments, they can align easily, while the shorter fragments actually they creates problem when there is repetitive sequences. So this is the advantage of the specific biosciences. But the problem with the specific biosciences is the error rate. Error rate is very high. So that is the bigger problem, also quite expensive as compared to the Illumina. The next sequencer is rather it's a Nanopore sequencing. So the Nanopore is actually based on the biological nanopores, which is generally used for the transporting the ions along with the membrane. So in this case again, you have to have the DNA fragments. You have to fragment them, and then you have to put the adapters, and then you have to do the sequencing. Again, in this case also the PCR step has been removed. So this is again the third generation sequencer, because the single molecule of the DNA you can sequence in this case. So what happens in this case in nanopore sequences, of course, as I mentioned, that there is a pore, small pore, which in general in the biological cell membrane, if you see, there are small pores, and these small pores actually they are used to transport the ions from the in and out of the cell. So these nanopores have been designed using the hemolysin with, of course, the modification. And what happens in this case is with the DNA fragment, with the DNA which you have prepared in your library, the one polymerase enzyme, or actually the one enzyme is attached to the DNA fragment. And this this enzyme actually also denatures the DNA, and it also pushes the DNA into this nanopore chamber. So once the DNA chamber moves inside this, DNA fragment moves inside this chamber, what happens is there is change in the voltage, change in the current happens. So as the DNA is moving inside, there will be change in the DNA, change in the current, and this change in the current is specific for a particular nucleotide. Let's say, suppose if A is passing through this chamber, then there will be change in the current, and that is specific to adenine only. When G is passing through that, then there is a specific change in the current to G or C or T. And also, even if the DNA is modified in the sense that if it is methylated, then that will also be a completely different way, different current, and that's how the sequencing happens. Means that if there is a change in the current, then that will be recorded, and the instrument, the algorithm knows that, okay, if this particular current is happening, it means this particular nucleotide has been passed through the chamber. The advantage of this nanopore sequence is, of course, it's in real time. Means as the sequencing is happening, you can see that exactly. Very long DNA fragments, up to 2 MB even, has been sequenced. But the limitation again is low throughput, means you cannot generate a lot of data, which you can generate in the Illumina sequencing, and quite high error rate is there in this. So now I'm coming to the Illumina sequencing. So I'm jumping directly onto the library preparation stuff. So again, in this case, library preparation, if you are doing an amplicon sequencing, means you are targeting a particular fragment of the DNA, let's say suppose 16S ribosomal RNA. So what you have to do is you have to design a specific primer which is complementary to your DNA, and with that you have to amplify your DNA fragment. Along with that primer specific to your sequences, you also add the Illumina sequencing primer in your primer sequences. So in the first step of the library preparation in the Illumina sequencing is you add the primer which is complementary to your target sequences. Also, you add the sequencing primer, and then you amplify. So what happens after the first PCR is you will have your target specific primer as well as the Illumina sequencing primers. In the next step, that is in the PCR2, what we do is we add the 8 nucleotide indices, and these 8 nucleotide indices are actually used to do for the multiplexing. You can combine many samples into one sequencing, which can be bioinformatically isolated after that. So you add the 8 nucleotide barcodes or indices, and also you add the P5 and P7 adapters. These are specific to the Illumina only. So we do the second PCR, and that's how the library preparation happens in the Illumina sequencing. Once the library is prepared, then this is the flow cell. I don't know if I have with me the flow cell. If everybody can see the flow cell. So this is the flow cell actually, Illumina flow cell for the MySeq. And this MySeq flow cell, actually this flow cell have the lawn of the oligonucleotides. And these oligonucleotides are complementary to the P7 adapters and the P5 adapters. So as in the previous slide, as I mentioned, that these are P5 and P7 adapters. And these P5 and P7 adapters are complementary to these nucleotide sequences which is there in your flow cell. So once you load your samples to the flow cell, what will happen is the sequencing primer will anneal to this, your fragment will anneal to the oligo, and the sequencing primer will anneal to your fragment of the DNA and starts doing the amplification, or you can say the polymerization in this case. So one by one nucleotides will get added and it will make a double-stranded DNA. After the double-stranded DNA forms, the original fragment of the DNA which came and bound to this P5 or P7 adapters will be discarded, and this newly synthesized DNA will falls back to the nearest oligonucleotide which is complementary, and then the sequencing, again the polymerization happens. This is actually called the bridge
amplification, because it is forming a bridge like U-shaped structure. So it is making a bridge. That's why it is called the bridge amplification. And this process continues for all the other fragments, and the millions of reads or millions of fragments will be generated. In this case, of course, so for the sequencing, what happens in the Sanger sequencing, of course, you know that this is modified dideoxynucleotide. In this case, we add the blocking group, which can be cleaved. Once it is cleaved, then the next step gets started. So again, one by one, the nucleotides will get added and your sequencing. So what happens is, first time your nucleotide will come. If it is complementary to that, it will get added, and then the terminator group, which is having the fluorescence group, also will be cleaved off, and after cleaving, it gives the fluorescence, and the fluorescence will be recorded by the CCD camera, which is under the flow cells. So next part is actually, I don't know how much of you are aware of the bioinformatics part, so I'm not going too much details into this, but the first thing is, once you get the data, first step you have to do the quality filtering. Means that whatever the wrong data you have get, that you have to remove. Then merge the pair and reads. It means that when the sequencing happens, you do the sequencing in the forward as well as the reverse side. So we have to merge those reads. And of course, the primers part, the primers which we have added in our previous steps, that we have to remove. We have to do the dereplication. We have to denoise. Denoise means, again, you have to remove all the bad data. And then finally, we have to generate the OTU table, operational taxonomic unit table. And then you have to blast those sequences to get the scientific name, and you can do all the other statistical analysis. So in this thing, this is specifically I have chosen for the meta barcoding analysis, which I did for my study. So for each study, there is a different kind of data analysis. There are different softwares. So don't be confused with this is the only way of doing the data analysis. There are many ways, and depending on what are your research questions, you have to do the different data analysis. So before going into the data analysis, what is the FASTQ format is? So you all know the FASTA format. So FASTA format is, of course, you have to have the greater than sign and the name of your sequence, whatever the name you have, and then ATGC. That's all. This is the FASTA format. All the next generation sequencers, they generate the data in the FASTQ format. So FASTQ is here for the quality. So FASTQ format actually comes in these 4 lines. For each sequence there are 4 lines. So the first line starts with the @ and it gives all the information of your sequencers, which sequencer you have used, which flow cell you have used, and everything. The next line is actually your sequences, A, T, G, and C. The third line is actually starts with the plus sign. You can have just one plus sign, or there might be some other information also. So this is actually the separator in one way. And the fourth line is your quality scores. So the quality scores in this case are ASCII values. So I'll explain this thing again. So the FASTQ format, there are 4 lines. You have to distinguish it from the FASTA format. In the FASTA formats, you have to just the greater than sign. All your nucleotides are there. In the FASTQ format, there are 4 lines. First line starts with the @ and then your information about your sequencer and everything. Then the next line is your nucleotide sequences. The third line is separator or identifier, which starts with the plus sign, and the fourth line is actually the quality score.
So the quality score is actually the ASCII format, ASCII format, ASCII. So ASCII format is American standard of coding for the information interchange. So like different sequences, they use the different Fred score or the quality score. So like for the Sanger, it uses the Fred plus 33, Illumina it uses plus 64. So different sequences, they use the different method of calculating the quality score. So let's say suppose if in your sequence there is J, this is the square bracket. So this, let's say suppose for the T there is a square bracket. It means the base quality, this T has a quality score of 29. How it has been calculated? So you can see the T. If you go over here, here is the T. So T comes somewhere around 100 and I don't know, maybe 73 comes I. Then let's say suppose it is whatever the value is there. Let's say suppose it is, if it is 100, so out of 100 you have to minus 64 if we are using the Illumina Fred 64 quality score. You have to minus that value and that will be your Fred quality score. Like if it is 104, if you sum of your nucleotides, if the quality score is H, so you have to minus 104 minus 64. So it will be 64 means 32. The value of that nucleotide will be 32. That will be the quality score of that nucleotide. And what this means of these quality scores are, if the value is, let's say suppose 10, it means there are chances of 1 error out of 10. If the quality score is 20, it means that there are chances of 1 error out of 100 nucleotides. If the quality score is 30, it means there is a chances of 1 error out of 1,000 nucleotides. If the quality score is 40, it means there is a chances of 1 error out of 10,000 nucleotides. So these are the way by which you can determine the quality of your DNA. So I don't know whether you could understand that or part or not, but this is the way that the quality is, that's how the quality is calculated. For each nucleotide, there is a quality, and it means what is the probability of that particular nucleotide, that this particular nucleotide is correct or it is wrong. So if the quality score of 29 or so it is almost 30, it means that there are chances that this particular T or thymine is, there are chances that out of 1,000 nucleotides, the chances of this is 1 time it will be a wrong nucleotide. So this is what the FASTQ format is. So first thing is, of course, you have to remove all the low quality score DNA fragments. As I mentioned that if the quality is if the quality score of some nucleotides are 10, it means there are chances 10% of the error rate, which is used in case of the sequencing. So of course we don't want that. Even if the quality score is 20, it means there is 1% of the error rate. That also we don't want. So we want all the wrong nucleotides to be discarded. So first thing what we do is we check our sequences, whether how good they looks like. So there is one FastQC. This is a program, software program, which is used to determine the quality score of your, or which is used to visualize actually your quality score. So if in this figure, if you see these bases after, let's say suppose after 22 bases, if you see these, they have very low quality score. I mean, if you see after 18 bases, they all have very low quality score, means below 20. So in our final analysis, data analysis, we don't want these kind of sequences. We want to get rid of this, and this first you have to visualize that what are the wrong sequences. So let's say, suppose if you want to get rid of all the sequences after 18 bases, then first you have to see the FastQ for you have to run this program FastQC, and the FastQC is actually a program which you can of course download on your computer in the Linux system, and then you have to put the output directory. Output directory means directory here is the folder. So you have to mention your name of the folder where you want to store the results, and then you have to give the file name where all your sequences are there. This is the coding for the running the FastQC. Once that FastQC is done, then you have to merge the paired end reads. So as I mentioned that in the Illumina sequencing, you sequence the forward reads and you also sequence the reverse read. And if, let's say suppose in this case, the forward reads is 8 base pair, reverse reads is also 8 base pairs. And they, these forward and the reverse read, they are also overlapping. So what we do, merge pair means we merge the forward and the reverse read and then make a single read. Read, as I mentioned previously, also the read here is just a DNA fragment. So for this, I have used the usearch program. So don't get confused, and I don't want you also that, okay, and also I don't expect that you will understand everything, because this is all hardcore bioinformatics. So there is a usearch program, and there are different options of the usearch program. So the first option is FastQ merge pairs. So what it does is it takes your FastQ files and it merges the forward and the reverse reads. And in this case, we mentioned that what is the minimum length we want. So anything which is less than 202 base pairs will be discarded, will not be merged. What is the minimum overlap? If the two forward and the reverse read, if they have the 100 base pair overlap, then only they will merge, otherwise they will be discarded. Then what is the maximum difference? Means what is the mismatches we want to allow. So if in the overlap sequences out of 100, if there are 3 mismatches, then it will merge those sequencing, otherwise it will discard those sequences. And then what is the output? We have whatever the name you want to give, you can give. So let's say suppose in this particular case, 8 sequences are there in forward, 8 are in the reverse, and they are like 1, 2, 3, 4, 5, 6 sequences are overlapping, and there is one mismatch. So if we say in this particular program, if we say that, okay, minimum fragment should be 7, minimum fragment length should be 7, so these 2 fragments will not be merged, it will be discarded. If we say minimum fragment length is 8, of course they will be merged. If we say that overlapping should be 6, of course they will merge. And if we say that there should not be any mismatch, then again they won't be merged and it will be discarded. And if we say that it should allow one mismatch, it will be merged, and you will get the merge sequences. So this is how you do for the pre-analysis, you do the filtering. Next step is, of course, you have to remove the primers. For the primer trimming, I have used the vSearch program. So vSearch program, in this case, I'm taking the merge sequences, which I did in the previous step, and then FastQ strip left. It means that what I am saying in this program is from the left-hand side, remove the 20 bases. So if your forward primer is 20 base pair, you have to say that, okay, remove from the left side 20 base pair. If the reverse
primer is 22 base pair, you can remove your 20 base pair from the right hand side, that is your reverse primer. So depending on your length of the primer, you can put these numbers. If it is 20 base pair, 18 base pair, 25 base pair, whatever the length is there, that you can remove, that you can put it over here. And then fast Q maximum error rate. Again, we are filtering that, okay, if in a sequence, out of 100 sequences, out of 100 nucleotides, 0.5 means if there is a 0.5% error is there, remove that read. Don't take into the final step. This is what I'm telling over here. And then finally, it is fast out. Finally, this is giving the output. So in this case, I'm removing the primers. The next step is the dereplication. So dereplication means is actually the removing the replicates. So when we do the sequences, millions of reads gets generated, and a single nucleotide, a single read can have many replicates of that. Let's say suppose in this case there are 8 different reads are there, but in Illumina sequencing and all, you get millions or even the billions of reads you get. So in this case, actually, let's say suppose 8 different reads are there, but actually there are only 3 unique reads. So what we do in this case is we tell the program that, okay, only take those reads which are unique and then give the numbers to those unique reads. Like in this case, read number 1 and 2, they are similar. Read number 3 is unique read. 4, 5, and 6 are exactly the same. 7 and 8 are exactly the same. So only 3 unique sequences are there. But sequence or read number 1 has how many are they? They are in number 2 are there. Read number 3, how many are they? Only 1. 4, 5, 6. So this particular one is how many are they are in number? 3 in number. 7 and 8, they are 2 in number. So over here, what this program does is it takes only the unique sequences and it records how many these reads are in actual. So it will assign this sequence as 2. It will assign this as 3 in number, and it will assign this as, let's say suppose it is in 4 in number or 2 in number. So this is what we do in the dereplication step. We remove the replication part, and we take only the unique sequences. And this is actually does to because if the millions of reads are there, it takes a lot of memory, and for the downstream analysis, it requires a lot of time. Then, if they are only the smaller number of sequences there, you can do the things very fast. Next step is denoising. So denoising is, you can say, the remove the noise. This is, the literal meaning of this is what it is. So what in this case is, again, in this case, we are removing all the error rates, what has been left even after filtering the different steps. So what we are saying in this case is, I have used in this case also the usage program. And what I'm saying in this case is, if a sequence is there in your file and the minimum size should be 5, it means what? If a particular sequence is there and if the number of those sequences are less than 5, then don't take into further analysis. This is what I'm saying. So in this way, what I'm telling to the program is that if only 1 or 2 sequences are there of a particular fragment, it means that might be because of some contamination, or it's just an error. So I don't want those kind of sequences. So what I have told to the program that, okay, minimum size should be at least 5. So this can be, of course, changed depending on what your data is and what are your research questions are. And then finally, we generate the OTU table, operational taxonomic unit.
So again, I have used the research and also the usage program. Both I have used. So as I mentioned previously, that for each sample, what we did was we did the dereplication. Dereplication means what? All the sequences which were unique, they were taken into account, and how many numbers of those unique sequences are there, they were recorded. So let's say, suppose for the OTU6, for the sample number F3, there are 749 sequences. For sample number, this one, there are 535 sequences. For this 313, this is how the OTU table gets generated, and that's how you get the abundance of each nucleotide or each read. And finally, we do the blasting. Blasting to get the scientific name of each of the reads. So let's say suppose for the OTU1, 100% matching has been done, and finally we get this is the genus and this is the species. For the OTU2, only 98% matching was done, and these species we get ESV4, we get this species. So over here, again, in this case, when I'm doing the blast and nucleotide blast, I have to put what is the database I'm using. So in this case, I use the NCBI data nucleotide database, and then the output format, which is the 6 output format, where I can get all the sequence ID, the taxonomic ID, scientific name, percent identity, what is the length, e-value, and the bit score. And percent identity, I have taken like 99%. It means that my sequence should match 99% minimum to the database sequences. And finally, of course, what are the different applications of the NGS? So there are numerous applications. In fact, in current scenario, current generation, this is called the genomic era, where everything can be sequenced, even like, as I mentioned that in my previous slides, that okay, initially when the human genome sequence started, it took 13 years to publish the whole genome sequencing of the human genome. Now, with our next generation sequencer, you can do all these things in just one day, not even one day, in few hours. So you can generate a lot of sequences, and those sequences are actually used in the medical field, where you can determine the different disease. You can measure the mutations. You know that even a single mutation can cause a disease. You must have heard about the sickle cell anemia, where just one mutation can cause the sickle cell anemia in a person. So the mutation can be checked through the NGS if you do the sequencing, because you can generate a lot of data. You can measure the genetic variation, because the different organisms, if they are staying in a different habitat, they will have completely different genome. And that is the reason why they are existing in the different habitat. And these genomes, actually, what are the genes responsible for these adaptation, you can check through the sequencing. Also, the transcriptome analysis. So transcriptome, actually, you know that for every phenotype, there is a gene expression, means there is a protein or there is a RNA expression is happening in your cell, and that finally translate into the phenotype. So with the NGS, you can do the transcriptome analysis, means RNA sequences. So in the previous generation of sequencers, we have to convert the RNA into the complementary DNA, and then we have to do the sequences. But now with our next generation sequencer, like third generation sequencer or the nanopore sequencing, you can directly sequence the RNA. Epigenome study, you know that epigenetic study is actually, if the DNA gets modified, there is no change in the nucleotide as such, but the nucleotide itself gets modified. That is, either it is methylated or some other, or whatever happens. So if it is methylated And that methylation pattern actually determines the regulatory mechanism in the genome. So if the nucleotide is methylated, it actually suppresses the gene expression. So by checking the methylation pattern, you can check the genetic modification in a particular genome. And these things can easily be done through the third generation sequencer. Even through the immunoprecipitation, like chip sequencing, can also be used. Another thing, of course, as I mentioned, that methylation pattern can be done. Environmental DNA barcoding means you can take the samples from the environment, and if you want to check what are the different species present in that environment, without knowing what are the different species present, you just sequence the DNA, and with the nucleotide databases, you can determine what are the different organisms present in that. Another application is, of course, the ancient DNA sequencing. Suppose if in the polar region, let's say suppose, or at a different depth in the sea, if you take the samples, you don't know what is there, because you know that layer by layer over the period of time, there is a deposition of layer. And if you are taking the samples from very deep in the ocean, it means it is the older one. So taking those samples, sequencing them, and determine what are the different species during that particular time. So that can also be done through the NGS, next generation sequencing. Of course, in the vaccine research, everybody knows that now in the pandemic time, the coronavirus, they are sequenced. Of course, they are sequenced through the NGS system only. So these are some of the list is on. There are too many applications. Almost everything can be done nowadays with the next generation sequencers. So this is the few. These are only the few list of the lists. And yeah, thank you. I'm ready to have questions. I don't know whether I'm able to convey all the message to you, but now I'm ready to take the questions.